Okay, welcome to the Classics in Immunology Journal Club. This week, we are going to do a couple of papers from 1970. Before I get started, I have to remember to tell you that if you like this journal club and if you like this particular video, don't forget to go on and like it and then click on the little bell so you can get notified when I do another one. And then also scroll down to the bottom and look at the other things on my website and social media with Twitter and so forth, Instagram. There's all kinds of things out there that you might enjoy. So the first paper that we're going to, so 1970, it turns out, was a banner year for immunology. And there was just an explosion of papers that were reported because what people had found was that they could separate T cells from B cells. And you have to remember that up until this time, we didn't talk about T cells or B cells. We knew that there were lymphocytes that were so-called thymic dependent that had resided in the thymus for some time. And then if you thymectomized a newborn mouse, that those cells just never developed. And they also knew that by this time that there were other cells that seemed to be the precursors of antibody forming cells, and which actually were plasma cells morphologically. But there wasn't such a thing as a T cell and a B cell yet, even though Max Cooper and Robert Good's group and also Jacques Miller had shown through in vivo experiments in the earlier 60s that there were these two different kinds of lymphocytes, probably. Functionally there were, but they couldn't really identify them. And so there was a young fellow by the name of Martin Raff who really hit the big time in spades. He happened to be a student with Av Mitchison at the National Institute of Medical Research in London. And Mitchison was a student of Sir Peter Medawar. There's a whole cadre of these people. And it turns out that Martin Raff was a young neurologist who was doing a stagiaire with Mitchison. And Mitchison had his fingers on the pulse of immunology at that particular point in time. And he knew that there was a particular molecule on the surface of all thymocytes, and it also looked like it was on the surface of a subset of spleen cells and lymph node cells. And he was able to obtain an antibody to this surface molecule, and the antibody was called, or the molecule was called theta, and it was anti-theta. And so this paper that was published in Nature in 1970 was one of several different papers that Martin Raff published in Nature and or Science and Immunology and so forth in 1970. He had like a half a dozen papers as a young student with Mitchison. The title of this paper is "The Role of Thymus-Derived Lymphocytes in Secondary Humoral Immune Responses in Mice." There was a tradition. This was when science was civilized, and it was really an old boys club essentially. Mm-hmm. And that's why we have all these single-authored papers. Mitchison, who was a student of Medawar, he had a whole string of single-author papers as a student, postdoc, and so forth and so on. So he's just passing the tradition on because now he's the professor and so forth. And what happened is that in 1980, a decade later, at least in the United States, they had woken up to the fact that you could make money from this biology stuff. And they passed the Bayh-Dole Act. Birch Bayh from Indiana, senator, and Senator Robert Dole from Kansas, Republican. Bayh was a Democrat, so it was a bipartisan situation. They established the ownership of any federally funded research with the grantee institution, as I've said in previous things. Well, that changed everything. You didn't see too many single author papers from students after that. So we're still in sort of the golden era of professors helping young people along that they thought had promise and so forth. And so Mitchison, he was looking for projects for his students and postdocs to do. And so he had this anti-Sera, anti-theta, and he gave it to Raff, and he said, check this out, see whether or not that this can recognize what's lymphocytes and so forth this antibody recognizes. And that led to a paper in Nature from Martin Raff in 1969, where he first published the first data indicating that this theta molecule antigen was present on thymocytes and also on thymic-derived lymphocytes. And then he published another paper in 1970 in Immunology, which was a greater exposition of this whole. And he really got a huge amount of attention from all of these papers that he published. And consequently, he made a career decision. I mean, he was a rock star as a kid, and he decided to become a scientist rather than a neurologist. He expanded on this in this particular paper. And in the introduction, he basically said because of what I just related to, he said, let's see whether or not this anti-theta molecule can be used to study the role of T cells or thymic-derived lymphocytes in the antibody forming response because of all these other in vivo experiments that had been done. So in these materials and methods, and of course, this is another one of these 2-page papers. So you can read these. There's a whole bunch of them on my website. And so you ought to go in there and check them out and glance over these guys. So in this instance, what they did was these were sort of an in vivo approach to life. He used what the immunologists call the hapten carrier system. Hapten being a small molecule attached to a bigger molecule, which is the carrier. And Baruch Benacerraf from NYU and Zoltan Overy had shown back in 1963 that you could get antibodies to the hapten as long as you had a big carrier molecule that you put the hapten on before you immunized. But that wasn't really, nobody really understood what was going on there. And that's what this paper is essentially about. And for the hapten, they used a small organic molecule the acronym is NIP, and they attached that to bovine serum albumin as the carrier, or they attached that to chicken gamma globulin. And so there's 2 different carriers and a single hapten in these. He would take this antigen, which is the hapten carrier, and inject it intraperitoneally into mice together he would precipitate the hapten carrier molecules with alum, and alum then became one of the favorite adjuvants in immunology. So he gave that to these mice intraperitoneally together with another adjuvant compound, whooping cough bacteria, pertussis. And so he was really hopping these mice up in this particular deal. And then he'd wait 2 to 3 months to get them so that they could generate this immune response, this mysterious immune response. And of course, that's what we're doing with the COVID vaccines now. We give them a single dose, and then we wait a month and give them another dose, a booster. So that's what Raff has done. And then he would kill these mice that had been immunized. He would take the spleen cells from these mice, and he would inject those spleen cells intraperitoneally into other mice as recipients that had been irradiated 12 hours previously. Total body radiation, 600 rads, which essentially stops any more lymphocyte formation within these animals. So the only lymphocytes they get from what he's put into them intraperitoneally. And so you could put the whole spleen cells into the animals, or he could treat the spleen cells, and this is from the previously immunized mouse, with anti-theta plus complement to lyse the spleen cells, to lyse whatever those cells were, and put those into the mice. Then he would wait 10 days and bleed the recipient mice, get blood usually from the tail vein, and he would then do an immunoprecipitation assay using ammonium sulfate that precipitates protein sort of non-specifically. When you get the right concentration of ammonium sulfate, you can bring down big molecules. And in this instance, what they had radiolabeled the hapten carrier conjugate, and so you could then separate the free from the precipitated with antibody, and that's that's called a radioimmunoprecipitation assay. And so it's a little bit more involved than the urinary plaque assay, but it allows you to look at this hapten carrier kind of a deal. And so in the results section of this paper, when everything worked great, they could get antibody that they could assay and they could detect and so forth. But if they killed off the theta bearing cells, they got no antibody or a marked decrease, greater than 80-90% decrease of antibodies. Furthermore, the cells that were giving the carrier effect, because they could compare chicken gamma globulin to bovine serum albumin as the carrier, those cells were the theta bearing cells. So that was, some of the first data that would indicate that the carrier is really due to these so-called what then became called T cells. So in the discussion of this paper, they came to the conclusion or the interpretation that the B cells that were the other splenocytes, they got help from the T cells, a little help from their friends. That was this was the birth of helper T cells. T cells help B cells. And as I've said before, because I'm a T cell person, the B cells didn't know what to do. They needed help. So the immunologic community started talking about this whole hapten carrier effect as B cells and T cells as thymic marrow synergism. So that was electrifying. And I remember, and so the next paper is also going to get into this, but before I go on to that one, I remember about a decade later, around 1980, I was riding on a bus at a meeting, at a conference. And I happened to be sitting next to this guy that was about my age and so forth. And we chatted, and we were talking about this era when they finally realized that there were T cells and B cells and they could separate them and identify them. He said, we all thought that was the end of immunology. There wasn't anything left to discover, because that just opened more things to discover. And in the beginning, and what that really did was, it was for the first time it was realized that there are subsets of cells, and that I'll get into a little bit later after I go through the second paper here. So this second paper that came out back to back with Martin Raff's paper, and this was in June of 1970, was from Germany, and it was from two authors this time, Annelise Schimpl Eberhard Wecker, and they were from the University of Würzburg, in just the northern part of Bavaria. Würzburg is about halfway between Frankfurt and Nuremberg. It's right in the middle of the Frankfurt Franconia wine country, and I've been there. It's very, very pretty, rolling hills, and so forth and so on. And Würzburg happens to be where Rudolf Virchow came from, and he was the father of cellular pathology. Around 1850, he decided that, up until that time, they were still working on theories coming out of the ancient Greeks that there were these bad humors that were causing diseases and so forth. But he knew on the basis when the microscope became invented and so forth that tissues were really made out of cells. And so he figured that, well, the diseases have got to be attributable to the cells as well. So this was Würzburg. And Annelise Schimpl was the student or the postdoc, and Eberhard Wecker was the professor at the University of Würzburg back in those days. So that set the stage for them to also look at this business between B cells and T cells. Donald Mosier, that we talked about a little bit earlier, in terms of saying that, well, you're showing that, okay, there's got to be macrophages are important also for antibody formation. And so the Schimpl and Wecker decided to use the Michel-Dutton system that we also just learned about a few weeks ago, to generate antibody forming cells in vitro. And what they did was they used thymocytes as their source of T cells, and they treated the splenocytes to put into the Michel-Dutton system. You remember, everything's in vitro. You take splenocytes, you put them in vitro, you try to immunize them with sheep red blood cells, and then you wait a few days, 4 or 5 days, and you assay them using the urinary plaque assay to look for antibody forming cells. Well, they could do the same thing that Raff had done. They could treat the spleen cells that they're going to use in these cultures with anti-theta, which is what they did, plus complement and try to kill off the so-called theta-bearing cells. And when they did that, just like Raff, they found that the anti-theta decreased the plaque-forming cell response by 80–90%. So they basically concluded that these results were consistent with the in vivo data that had been published, and they supported the idea, the notion that you needed T cell and B cells to cooperate with one another in order to generate antibodies. So then they go on and this is also another of these 2-page papers, 1 or 2-page papers. So there's not a lot of things in these papers, but the question became then, and so, well, what are the T cells doing? How do they help the B cells? How does that happen? And as I said, this was the beginning of the whole idea that even though all the lymphocytes under the microscope look exactly the same, B cells and T cells, and they're all small, round lymphocytes without much cytoplasm, just mostly nucleus. But if you could separate them on the basis of their surface molecules, this was very, very, very important. And it was only a couple of years later then that Len Herzenberg from Stanford, we're not going to review this paper, but you can look it up online. In 1972, he published a paper that was very similar to both of these papers, but with the idea that we needed a better way to separate these cells. And so he took advantage of a new machine that had been generated by the physicists in cell biology. They got involved in cell biology. And he told me, I was out visiting him, and I said, well, how did you get into this? So he got into the fluorescence activated cell sorter, or the fax machine, FACS. It wasn't FAX, it was FACS. The fax machine. And what that machine would do is that you could, instead of having used theta and complement to kill off the thymocytes, or you could also then have antibodies to antibodies, anti-immunoglobulin. You could take these, and those would recognize B cells, and this is what the first paper from Herzenberg's lab is all about. You could separate them in the fax machine. You put your sample into this machine, and it would form a stream of fluid that each drop had a single cell, and they would drop down, and then you'd have a laser beam that would hit that cell. You had to have previously conjugated your antibodies with a fluorescent molecule. So when the laser hit it, it would add energy to it, and then with a magnet you could deflect it into a separate pot. And Herzenberg showed that he could purify B cells versus the T cells 500-fold using this machine. Well, that was the beginning of all kinds of things. The fax machine or the flow cytometer essentially supplanted it. I mean, if you didn't use the flow cytometer, you weren't really doing real immunology. However, they basically built their own machine at Stanford, and it took up a whole room. You know, it's like those stories about the first computers, and they take up a whole room. It's exactly the same thing, and you needed a physicist to be able to run the thing. You couldn't do it yourself because the thing would always go snafuing out and so forth and so on. And it took about a decade for more and more people to get fax machines. But by the 1980s, that was all, and even now. So now this is the 2021s. Flow cytometry is where it's at in immunology, and separating cells from one another on the basis of all kinds of surface markers, and then trying to understand what their functional capacities are, capabilities are. That's what immunology became. So I'll stop there, and don't forget to like this video and click on the bell, and look for everything else on my website, and particularly all the papers from 1970 that you'll find there together with these two papers. So thanks very much. Thanks, great.